If anybody want to ask questions or any discussion, anything, so please use the microphone. Without that, I think you will not be able to get the signal. Okay. I think what we're gonna do, the tutorial from the morning, we just do it from the do the morning tutorial one. I think the one from the yesterday that I gave you talk, afternoon talk, it's okay. It's very general, and we can do the tutorial anytime that we need. That's from the yesterday's tutorial 2. That one is just a very general tutorial. I think we are okay. So I think important is that the afternoon talk that I gave you, I think it was important for you more than the tutorial that from the talk, right? That was missing from yesterday. So whatever is missing from today, if anything, afternoon session, therapeutic or anything, we can cover it someday. We have still have 3 days, so we can cover it. And also I will squeeze it in somewhere, some other during while I'm giving other talks, we can squeeze it in somewhere, and we hint it so that you will definitely will be fine. I can cover it here and there. Okay, that's not a problem. So the today's journey, I think the morning, that journey I was talking about from biology, preclinical studies to the clinical study, right? So there are a lot of questions I think you circulated. It's just we covered all of the talks that we gave. I think couple of questions, but it's okay if you can understand it. It's okay if you don't also. It's fine. It's just that if you can understand and if you have questions, I'm okay to answer, okay? If you cannot, if you will not be, it's fine too. But I think overall design, that the way that we designed, so that you will understand what it takes to get into, preclinical studies to translational to a clinical development, right? So we just want to hit that and give you a hint that any study that you do that should have some benefit for somewhere, something, right? We all do research without benefit also, many times, happens for many reasons. Also, same time, so you have to have something that actually gives something therapeutic value or understanding the biology, which actually helps with some medicine. Or that is, we are biologists. We have to really bring some medicine to the clinic. The clinicians are not going to bring the medicine. We are the one giving the medicine to them. So that they treat the patient. That's how we need to think, right? That's why this research is important, and conducting the right experiment is important. So the morning session on therapeutic resistance, and also why combination therapy important and necessary for pancreatic cancer. We taken the pancreatic cancer as an example. It's the same thing for anything. So many of you probably worked on different resistance pathway and different resistance in tumor cells, different tumor, things like that, or maybe non-tumor also. There is a lot of resistance pathway they work on too. But I think important is that whether you understood the concept, the design, it's fine if you don't. But so the first question is, why is the combination therapy approach is necessary for pancreatic cancer, right? I was actually telling you while presenting that there is a pancreatic cancer driven by multiple, redundant signaling pathway, right? And there is a dense stroma, immunosuppressive stroma that we came to know later. It's a immunosuppressive stroma, and the single agent therapy, right? I was talking about the SARC, typically fail due to compensatory activation. Like when you inhibit SARC, something else getting activated, like STAT3 or something else is activated. What you have not seen is that, what I have not showed you is that many other signaling also get activated, right? But you have to focus on something that actually it is not that only those signaling pathway. Something else maybe might have happened. Maybe I did not show you, right? Maybe it is there, maybe it did not. Maybe happened opposite. Okay, there are many things happen, but in the research, I think important is that take the one which actually makes sense to you and can bring the data to and also can have a therapeutic value to it. That's how it is. So it is not that we have conducted only those signaling pathways and only you are showing. It's just a published signaling pathways and it has a meaning into it and also a value to it, and also it is translatable, right? Also we showed that alternative pathway, that is a STAT3 activation. When SARC and our MEC was inhibited, STAT3 was getting activated. So that's why we did the combination therapy. We knew that the combination therapy is the thing that we had to do, but when we started, it is not, right? We started with a single therapy and went into combination therapy, right? That is that question. And there is a two major factors contributing to therapeutic resistance. I think you already know one is stroma. Stroma is, I think those who are working in cancer, they all know stroma is one of the resistant pathway. We also hit at the same thing. In our work also, the stroma came in between the fibrotic and poorly vascularized microenvironment. So that actually, if you saw the data, the drug delivery data that you saw, mass spec analysis and all those, you saw all those data. So many things that at that time we did not do is a immune, study, right? So that doesn't mean that we are not doing now. So we may be doing now, right? So means that we never stop. If the technology changes and something else comes in the field, if someone says the stem cell is important, that is the hot area, probably we're going to look into stem cells too. So because we are not going to stop because we are done with it, we published the paper. No, we're going to continue and establish that area, understand more, and study. That's what it is. The resistant pathway, it is, oh okay, that means that we came out of resistance and we are done with the research. No, there may be other resistance, there may be other factors involved. So that's why we need to keep on understanding, and that's how we understand the biology, right? Means that you do something else will happen, understand that, and something else will happen, and then understand that, and then you add that and delete, and then keep on working, understand more and more, and the knowledge you create and the biology you create, I think is huge, right? So, but the passion, you have to have the passion towards it. Then only I think you can do all these things. And also for the major, two major factors is the other one is mutational burden, right? Mutation burden is a key factor. So whenever we modeled the mice, genetic mouse model, I did not go all this modeling and all those things. We use the genetic mouse model which has KRAS mutation, P53 mutation, or sometime we use the TGF beta receptor knockout. I did not talk about all this. It's like we modeled such a way that mice was already there. The genetic mouse model, someone else developed those mice and we use it. So these are the two. One is stroma, another one is a mutational burden. So next question was, how does the STAT3 become activated downstream of SARC and EGFR, and why is this important in pancreatic cancer? So you already know the SARC and EGFR activation leading to STAT3, which then translate into nucleus. Once the STAT3 getting activated, it will go to the nucleus. So STAT3 is a transcription factor. So it go to nucleus and drive the transcription regulation. The STAT3 regulates IL-6. There are a lot of other factors that regulates. Okay, if anything that going into the nucleus, right, phosphorylate and then going into the nucleus. Remember, when you are studying anything signaling pathway, you have to understand that what phosphorylates, what not phosphorylates, right? And when they phosphorylate, where that is going, whether going to the nucleus, you have to visualize those things though. You have to do the confocal or regular imaging, anything, microscopy you use with the color microscopies, you can actually see it, all those things. It's very easy. And anything that you conduct the experiment, try to see it from your eye and see whether what you are doing and what you are thinking is happening in your model, right? So that we saw the STAT3 was translocated to the nucleus and drive that lot of other transcription factor that I did not show you. Okay, all those gene transcription, regulation, I didn't want to bore you in telling all the regulatory pathways and how it is happening. Rather than that, I want to give you a kind of a motivation to study and understand why you want to study and all those things. In pancreatic cancer, I also told you that STAT3 is hyperactive and supports the survival and proliferation. Now STAT3 is a well-known regulator. Like people study. Even today, there was a paper in the morning that I saw. Just saw there's a STAT3 signaling. Very high impact paper. So just a common, but they always try to find something new, because you should not stop thinking about it. For example, I will give you a story. Someone told me, don't work on the EGFR because the drug is okay, but there's a lot of other problems. There are a lot of other issues, right? And today, if you see, we were thinking about something, that same thing, there is a publication came out saying that EGFR activation is important for the stroma. Okay, there was a paper came out in the pancreatic cancer. So what I'm trying to tell you is that if someone stops you, oh, okay, that may not work, this may not work, and same with leader, it doesn't matter what the others say. You think yourself that what is important, whether what you're seeing is important, whether you see it yourself. In the research, all you have to do is that see it yourself. Doesn't matter who says what. See it yourself. Then only you can actually design the next experiment. If you don't see it, you will not be able to design. You're actually doing someone else's saying to you. Even though your mentor says that, oh yeah, it works fine, go ahead and do it. No, don't do it. You see yourself. You're already a scientist when you sign up for a PhD. You don't need a mentor. Okay? You are already scientist. You just have to do it. If your mentor says according to their experience, how do you know whether it is right or wrong? You never know. You figure it out because you are also already scientists, okay? That's how you need to think if you want to conduct research, right? If you want to enroll in the PhD program, this is how you need to think. If you have done PhD, still keep thinking, because you're not done yet, because you have a bigger problem than doing the PhD, because now you're entering to the scientific world where you are a doctor already. So you have more things to work on. So next question is, what role do large-scale genomic database like TCGA play identifying therapeutic target? A big way. It's a big way, because if you can understand TCGA database system, it's huge. You can actually Google it and see TCGA, and there is a lot of information. For that, whether you have time to understand it, right? You need to have another partner who actually knows already to understand how to do it. Otherwise, you have to spend all day and night, so many months to understand it, and you don't know, end of the day, you actually get the exciting data, but you don't know whether it is true or not, whether I should be using it or not. Someone has to give you a confidence, yeah, go ahead and use it. What is the problem? This is actually right. So someone has to give you a confidence, right? So you need a partner who actually understands the science. Means that a smart scientist around you is also important. If you are smart, bring another smarter. Bring otherwise some duplicate person, right? It's okay. If he says yes also, it's okay, fine. I'll give you motivation and energy. So important is that having a partner in the research is a key factor. Someone to talk about frustration. Someone to talk about your just whatever the results you got. Someone to listen to you, right? Someone to listen to you all the time, who actually listen to you and say something. It's okay, fine. But it's important having a partner, right partner in the research, also a key factor, right? And the next question was, why SRC kinase is considered a gateway for resistance mechanism in pancreatic cancer? It interferes with multiple, the SRC interferes with multiple receptor tyrosine kinase. There was an investigator in Vanderbilt when I started. They were working on all those things. That's how I started. But nowadays there's a lot of things, information available. But why we selected SRC? Because we knew it is actually interfere with many other signaling pathways. That's why I started with that. And also I knew there is a person who actually next door with whom I can actually discuss and talk, and there is a postdoc can easily give out all the information from the lab. Mentor may not give, but a postdoc or the graduate student, PhD student can give out everything what mentor is doing. So I knew kind of all this information will be collected, and it's important for science. Science is not for me, it's for the human, some humanity. Is just for everyone. So that's how you need to think. If you're taking information from someone, it is not for you. You basically, end of the day, you're going to publish it, and publish it in the right way, but also give the credit to the person who actually give you information directly or indirectly. So always credit them, and it is not yours. The work is maybe I published it. It is not mine. It is now in the public, right? I got the money from the NIH to work on this. That NIH is a public database. That is giving out to the people. So it's for the people we are working for, right? So it is not for me. So it is just that you have to think. That if you're getting the information, that doesn't mean that it is yours. You publish it, that's all. Probably you may think that that is yours, but it is not yours. Once you publish, it's public. Okay? Until you don't publish, it's yours. Once you publish it, it's for public. So it's not yours anymore. So if you read the copyright from the journals, you will know that is not yours. It is for the journal. Journal is taken over. And journal to a public, when the NIH will take over in NIH in the US. But here, you know how the thing policies work. It is like that. So always you think about whatever you're working in science is for public. But whatever your job and you're getting the salary and things, that's all you got. So rest is for public. But it doesn't matter what you say. And what was the translation? I think that was describe a key finding from preclinical studies that combine SARC and EGFR inhibition with gemcitabine. See, this is a triple combination, right? SARC, EGFR, and chemotherapy we did. See, we didn't know, we designed every sort of things, but only given you a data which actually worked out for us. But the science worked out for us. But a lot of frustration there, right? When you're doing the conducting research, there's a lot of frustration there, right? We did not work. So those kind of data probably we are not showing you. But important is that the triple combination suppress the activated STAT3, and then it did not do in a single agent or the double agent. So triple agent worked out very well. So this led into many other functional issue. In vivo tumor growth was reduced and stuff. But important is for us, highlighting part was the STAT3, right? We got the STAT3. That is a highlight. When you're studying, when you're doing anything, right? You may be doing this and that. End of the day, what you got is the key thing. You get something, but you should be having awareness to know that whether that is important or not. What you got, right? I think that is the key part. If you can actually realize and understand what you got is important or not, I think that's enough. That itself is huge. That actually takes your whole life to figure it out what it happens. For example, IF1 alpha that you was talking about, though you targeted IF1 alpha worked out, and now understanding the IF1 alpha itself is a biology. It's a huge biology. It may have a role in different things, just not in the cancer. Many other, for different cell types. There were IF1 alpha, maybe they're in the T cell, and there some other cell type. Things like that. You have to discover those. Just don't stop it. So don't jump on to next time AKT or some other signaling pathway. Just stick on to something. Try to figure it out. What's going to happen? Even though you fail, but there is no fail in the science. Okay. Whatever you get, whether it's a negative or the positive, you always learn. But what I'm saying is the fail means that you fail figuring it out where this is hitting. It's okay. But you still publish whatever you have done, that wherever you went, right? So the rationale behind the use of testing dasatinib and erlotinib, the SARC inhibitor, EGFR inhibitor with gemcitabine in a phase 1 trial. What was the rationale, right? So the rationale was the evidence we got are showed that SARC, EGFR inhibition, feedback loop driving the STAT3 activation. And also it enhances the chemotherapy delivery. So you saw in the mass spec analysis chemotherapy delivery. That actually huge. So when the drug is going into the tumor, that itself is actually huge. We got the data later on, but after the trial, but still it is huge, and we wanted to study. So those kind of things. And the next question is, how does inhibiting STAT3 via SRC EGFR blockade improves chemotherapy delivery to the tumor? So inhibiting STAT3 can, normalize the tumor vasculature that I showed you. Also, it reduces the interstitial fluid pressure, those kind of things. So important, I think, also finally we showed in the tumor mass spec analysis, when you inhibit STAT3, what happens to the drug? The drug delivery to the tumor was much better, as those kind of things. So next is, why is STAT3 considered a central mediator of tumor stroma interaction in PDAC? So STAT3 promotes, pro-fibrotic and immunosuppressive factors that, that sustains the dysplasia and the stromal area, and also it blocks the immune responses. So we kind of knew that kind of research already, people were started publishing around that angle. So this microenvironment study that both protects the tumor cells and also therapeutic, whatever that we have studied, I think helped us to understand even more. So next was, what was the outcome of combining STAT inhibitor AZD1480 with gemcitabine in PDAC? So you already know all that, the whole data. The combination led to improved the vascularization, enhanced the drug delivery I just mentioned. We saw the drug delivery into the tumor and resulting in reduced tumor growth and also prolonged survival. So prolonged survival is also key factor. I think, I don't know whether I showed you, I think I showed you in one of the figure. I think it's not about STAT3, SRC, EGFR, whatever I'm talking about. It's just I've taken a concept idea for you to tell the story. Important is that, I might have done the mass spec analysis to see the drug delivery. You may do something else. Okay, it's just that, if you don't have the mass spec, facility, you will figure it out something else, that how you want to see the drug delivery. You have to find out though. Or if you're in your study, it may not be drug delivery, it may be something else. But what I'm trying to tell you is that important is that whatever the problem is, that you think about it and study that problem and figure it out, that how do you present the data as a data on your paper as a picture. Okay, that's a picture. I wanted a picture actually shows the drug is delivered, because I might say in a dot plot or some other figure, but picture actually for me is important. As a scientist, I want to see it, right? That's why I went and did the mass spec analysis. Probably you may not have the facility, you may have to do something else. So in your area of research, you have to find how you want to show a picture. For example, somebody was saying something cell issue. So take a picture and present it such a way that see the difference between the different cells from different company and whatever that is. But picture makes the big difference whenever you are trying to establish and try to show comparison between different types, right? Are delivered or not delivered, right? Okay. Summarize how STAT3 inhibition can help overcome chemo resistance. I think PDAC. I think I already summarized. It's when you inhibit STAT3, it reduces tumor promoting cytokines growth signal, decreases the fibrosis, and I also showed you vascular normalization and all. Those are all the things. And also next question was on what role does TASE or ADAM17 play a mediating resistance when STAT3 and MEK inhibited? So the TASE or ADAM17, EGFR, and cleaving, TASE is cleaving the amphiregulin, pro-EGFR ligand it's called, and ligand is going and binding on the EGFR and activating the downstream signaling. These are all story, okay, in my work. But that story in your work is maybe different, but it's a similar way, okay. You may be studying different receptor, you're studying different ligand, you're studying different signaling pathway, but the story is same. What I'm trying to tell you is that you had to build the story that actually is real in your study, so that actually becomes a scientific story for you later on when you are establishing your paper or figure or anything, right? Um, MEK and STAT3, why do MEK inhibitors alone often fail in pancreatic cancer? Remember, blocking the ERK-MEK pathway can also trigger the reciprocal activation of STAT3. That's what I showed you. And also vice versa. When you inhibit STAT3, the MEK actually getting activated. This feedback loop allows tumor cells to maintain survival signal, right? When the STAT3 is activated, when you inhibit MEK, STAT3 is also survival signal. Actually, when it increases, tumor is actually increases, proliferation is increases. And when you inhibit STAT3, MEK is activated. That means that is also pro-survival signaling. So survival is not going to happen. That's the loop that we understood. That's why we wanted to block both, but it did not work for us. But what I'm saying is that not the way that we were thinking, but it can happen. But for good, end of the day, we found out through immunotherapy, right? How does this dual MEK STAT3 blockade affect the CAFs and immune cells? I think I did not touch the base on the CAFs. CAFs are very important. That is a cancer-associated fibroblasts. So very important study. There are a lot of papers coming out. If you see CAFs, there are a lot of great papers are coming out nowadays. And the people think that CAFs important to study CAF cells and also immune cells to increase your impact. This combination therapy can shift the CAFs towards less pro-tumorigenic phenotype. It increases the intratumoral T cell trafficking. I think overall what I wanted to tell you is that it promotes immune active microenvironment. That's I think the important study for the immunotherapy. So what clinical outcome was observed in a patient with metastatic chemotherapy refractory PDAC treated with MEK STAT3 PD1 blockade? I think the patient showed notable reduction in both the size and metabolic activity of the tumor. Providing, I did not cover all those things, but I think they provided some clinical proof of concept that triple combination really can overcome the resistance in pancreatic cancer. I think that's all I can tell you. What is the significance of this trial, the phase 1 and the triple combination in advanced pancreatic cancer? It's a next generation therapeutic strategy. You know what I'm saying? We don't stop there though. Okay, the strategy is okay. When we did the SARC and EGFR chemotherapy, at that time, that itself is great. This time, this is great because we added immunotherapy, and then the field is also going to the immunotherapy. The field is going to the immunotherapy. The reason field is going, they see something is important that we are missing, right? That's how it is going. And we also went with them, and I think it worked out, right? And also, when next time, if you see, probably we'll be doing something else, which is actually important. The strategy may be next strategy may be different, and maybe we'll be doing that. That's why the literature, reading literature, understanding what is going on, present world, outside, is important. So because you have to change the strategy. You are a scientist and also you are an industrialist. So you should not forget, you have to think like a corporate. Also, if you want to establish your science, publish a paper, you have to think like corporate. It means that think like that anything can change tomorrow. So you have to update yourself tomorrow. There may be a literature coming in the morning. So you have to update, make sure in your area, whatever is published, you're actually having that up-to-date information so that you can actually learn from it. But we can actually, I have subscribed to Gmail. Anything that I'm interested, it'll pop up and come to my Gmail so that I always see those papers and just to glance. Okay, so it's important to have that keep coming to your email so that you're not forgetting, right? So summarize the integrated mechanistic model proposed for PDAC therapy. So I think the model I highlighted, like SARC, EGFR, and STAT3, interconnecting with the MEK-ERK pathway, and how this is influencing the tumor microenvironment. These are all the effective therapy, right? It's a blockade. There's independently also these signaling pathways can work. But it worked. That's, I think, the key factor. Okay, so what are potential future directions and all those things? So there are a lot of biomarker development that we are doing. So I think immunotherapy combination worked. So we are moving forward to personalized approach. We can actually do mutational profiling, stromal profiling. I mean, clinical trial can be larger. If this is working, then we should make it like in a larger. Also, we are looking into different angle. What else is missing? There's always research going on in the laboratory. Like, what else is missing? So my direction is also CREB signaling pathway and all those things. So I don't have a drug like STAT3 MEK, because they have the companies they're developing. CREB is still growing, is a baby. So there are not many drugs available. So I'm also struggling to make the drug. I'm also working with a lot of companies to get the good drug out of it, so that I can actually target. My ideas of CREB is entirely different concept ideas, and this is entirely different, and approach is different. So like that. So we keep thinking both the angle. We learn and then add another angle to our research. That's how we do. So you may have one research going on in your laboratory. You may be conducting one type of research in your laboratory, or you are planning one type of research in your laboratory. There is always, we say, a plan B. Like in your life, you have a plan B, right? Because you may fail in this research and have a plan B, and your life is better. Like plan B means that always have a publication type of work that you actually get the publication. Otherwise, I will not be able to survive as a scientist. I will not be moved forward, because if I'm actually thinking about the humanity completely, probably I may fail in my own research and survival, right? So humanity is important if I am surviving in my academic environment, right? For that academia, you have to publish paper. You have to get a grant. So you have to think the side by side, how do I survive in academia as a move forward, so that I can spend more time and serve my humanity, whatever that is I'm thinking. So if you are not existing in this world as an academician, you will not be able to do anything much. So you have survival. For that survival, you have to have a plan B, plan C, plan D. Because it's very important, publishing a right, another one, which is actually easy to publish. Like I was telling you about TCGA data and things like that, bioinformatics and collaborating with someone, and maybe you're expert in something, and maybe you're giving idea. You have to, because I said expert, you have to be expert with something. Okay, you have to learn. Go do the workshop, get some idea and become expert. Say something that I'm an expert in cellular signaling, I say sometimes. But now I'm changing. I'm actually expert in something else, right? You have to say one day that I'm expert in this. What are you expert in? Like for example, a mechanical engineer, expert in many things, AutoCAD or whatever that, tire changing, whatever that is, or engine or whatever that is, right? He's expert in something. So in biology also, you're expert in something. You had to find a way. If you are not expert, right, become one. You want to sell, become one. That's why, how do you become one? Because you have to go to the workshop and go to learn all those things. Okay, you want to become a bioinformatician? Please do. Go ahead and learn all this. If you have a knowledge of computer science, or you are maybe better, you are a biologist for maybe you came because it's easy, but you may have a brain of computer science. Learn something bioinformatics, learn something software. Probably it help you and also help others, and you become a great bioinformatician. Maybe as a computational biologist, maybe helps you a lot of investigators. Or if you like immunology, start learning today. Go to workshops, go to symposia, go to seminar, go to other laboratory, immunology laboratory, start learning, reading, things like that. And one day you'll become immunologist. I'm telling you. You start, whenever you think you want to become something, start that day. You'll become one day that one. So it is like that. So it may take some time, but you'll become. So it's like that. So in science, in biology, you have to become one expert of something. It's important for your survival, right? So expert of something, don't be general. So general is a key thing, but also expert in one particular thing is also important, because you're contributing something to the society, something to the another investigator, something you're giving out to that your expertise, so that helps other people also, and also you are expert so that you get publication, co-author paper, and people call you and give you a presentation and talks and all those things. It's also important, giving out something out for the society. So that's where we are. And now it's open for questions if anybody has.
Sir, when we can decide that we have to go from preclinical to clinical trials?
When you have to decide.
Yes.
So you want to go from the preclinical to clinical. You don't decide. It should happen. Huh? No. I told you the story that clinicians came, found out that this is important for the development, right? Sometimes you can also go, if you think this is really translatable, right? You have to develop that type of knowledge, that clinical development knowledge. Okay, my work is ready. So as a biologist, you have to think also like a clinical development. If you don't develop, like for example, you have to understand what it takes to, take your work to a clinical development. Or you have to know all the phase one, how that works. There is a huge knowledge out there, right? You have to know all those things before you even decide your work can go there, right? It's about that. Then you don't need anyone. You can decide yourself. So your work, whether this work will go or not, you can even design your work such a way that you can develop the preclinical development so that your work can be translatable. Okay, if you can understand, okay, if we can you can understand the preclinical development, how it works. So I think the knowledge that you develop in the clinical development can help you to design the right experiment so that you can take your work to the clinical. Yeah.
So when you were using two combinations or three combinations of drug, in the control animals, did you find any toxicity issues?
So we always do the normal mice, okay, and make sure that toxicity is not there, right? So when we do the drug study, we also do on the normal cell line. And when we are doing the signaling pathway, I did not show you in the normal cell line. I think in one of the molecular cancer therapeutics, I think I show you later in another study that when I'm present tomorrow, is that we actually checked on the normal cells too. Any drug you study, you have to check on the normal cells, make sure. And sometimes the toxicity is high. You have to talk to the medical oncologist and see whether this much of toxicity is considered okay or not. So when it comes to toxicity, right? They grade in the clinic that's why I was telling you, you have to develop the knowledge of clinical development. If you don't have the knowledge, you will not know how to do your conduct. So in the in the patient, you have a grade 1, grade 2, grade 3 toxicity. So grade 1 and grade 2 is okay if you have in the grade 1 and grade 2. Grade 3 toxicity, you cannot continue that work, probably toxicity, too much of toxicity. So like that, when you are grading it, that you analyze some of these factors, so that's how you grade them, right? So there's same concept design, you can do it in your in vivo mice also. So same grade 1, grade 2, grade 3, your own way. right? That knowledge, you should not have that lacking. You have to, if you want to really do something, right? Develop the knowledge so that awareness, read those papers. We as a scientists, right, PhDs, what we do, we don't read those. Okay, we think that it is for someone. That is where we lack. I think I was there like same way, right? When I realize that I'm lacking in something, I'm actually going to someone, I'm hiding somewhere when someone is talking about toxicity. When someone is talking about the patient data, I'm hiding. Okay, it is not me. It's not SARC or EGFR. I'm not worried. No, it's very important. But very important because I'm not able to understand. That's why I'm running away. But you have to try. You have to be there. You have to understand. That's the knowledge. So don't run away. If you're running away, that means that you're losing real science for your work, right? So what I'm saying that you have to conduct the normal human, mice without tumor and make sure the toxicity is not that high. And also normal cell line, you have to run the normal cell line. When you're conducting your own experiment, when you're doing the resistance, non-resistance, and also just run the cell. If you have the normal cell, I think important to run. If you don't have a normal cell, it's okay. If you have a pre-neoplasia cells, is also fine. You can run too, right? But it's easy in conducting in the normal mice, right? Without any tumor. Yeah.
One more question.
Yeah.
When you were talking about chemo resistance, you have chosen stroma as one of the factor. Have you seen any things with respect to stemness markers, because those are also causing resistance?
Yes, you're absolutely right. There's a lot of work on stemness. My lab did not work on the stem cell area. We did try to go enter that stem cell area, but we did not. So there is definitely a stem cell hub is another angle and area, which is actually important also. There is a lot of work, a lot of target they have developed, and there are a lot of stem cell marker. For example, if you take STAT3, there is pancreatic cancer, there is a stem cell marker specific to pancreatic cancer. The same way you have other cancers, specific stem cell markers too developed already. Huge data out there. So they keep coming. Okay, every other week you see there's a new paper. Doesn't matter which area you're going into. So hopefully that will help you. Yeah, stem cell is a great area. But important is that doing it right. Doing the right marker screening, I think, a key factor. Say, for example, there is no better or the worse. It's all about what's happening in the mice or in vivo when you do the combination. One thing is our research is taking us there. So we are not going there, right? So your research, your work should take there. Doesn't matter how many combination you're going to do. Your research actually, should take you there. For example, I did SARC, I did the therapy, but then I found out what happened and then added EGFR inhibitor. Then that was not enough. I had to add the chemotherapy. Chemotherapy is standard therapy. Then clinicians were told me, if you want to do the trial for this, you have to have the chemotherapy to it. Otherwise, nobody is going to do it. And also, I wanted to publish in a clinical cancer research. It was a paper from the ACR. So to publish there, the clinical cancer research, that means that only clinical related papers will be published there. And also a lot of clinicians watch those papers and see those papers, read those papers. So that is also another important thing that you need to think that where you want to publish. If I want to publish that paper in molecular cancer therapeutics, I would have published just that, SARC and EGFR. But no, I want to publish in clinical cancer research so that mine should be translatable. So that's why I did the clinical the triple combination, so that it's so translatable, and actually we did translation. And the same way STAT3 we did, and it did not go to the clinical trial, right? Because we did not do the way that did not happen. The survival was not that great, and things like that. And when we did the MEK inhibitor together with STAT3, actually our work took us there. The STAT3 inhibitor activated MEK, vice versa. So our work took all the way to that. And the SARC and EGFR, MEK and STAT3, I published in Cancer Research. Cancer Research is a, kind of a, they don't care about this clinical development at that time. Now they are huge in clinical development too, but not that way, the like clinical cancer research paper or cancer discovery. We published in cancer discovery also, co-authored and things like that. But that's, discovery is different. When you say discovery, that means that it's a huge discovery you might have done, right? But cancer research has a mechanistic approach you have to give. That is cancer research paper. So you have to know where you want to publish your paper. It is a key factor, right? If it is cancer research, they like mechanism. That's why the TASE, EGFR, and all those things, the mechanistic approach to our work, our study, we studied all those things to publish and then went into a cancer research paper. So then we were thinking, we were lacking in a lot of things, right, during that time. Then we were thinking and added the PD-1 therapy. What I'm trying to say that it all depends on your work, where you want to publish, and what you want to do with the work. It's about that. Yeah, you can do, there is a study they have conducted where they can actually tag with the chemotherapy something and deliver to the drug, drug to the tumor site, right? Because pancreatic cancer tumor was a harder tumor, the delivery through something else, another vehicle is hard because it may not go through. It's hard to go through. So in some other cancer, people have done a lot of delivery thing, that how you are actually talking about. It was a hard tumor, so it was difficult to do the nanoparticle delivery, and like chemotherapy they do change something and then chemically and deliver those drugs to that site and all those things. Yeah, you can do a lot of things. It also depends on the cancer. Yeah. Yes, yes. Okay. Okay, thank you so much, everyone. I think we had a fantastic day. I know you are all tired and want to go back home. So we will have tomorrow, same time, I think, 10 o'clock. I think we had a great session. If anything, any questions, any general question, please come and see me, and if you want to discuss anything, happy to have a few minutes. So otherwise, thanks everyone, and have a great evening.
Good evening, everyone. I would like to thank Dr. Nagaraj Nagattihalli and Professor Chandrasekhar Joshi for their lectures today. This marks the end of day 2. Thank you, everyone. Have a nice day.